Hey guys, welcome back. Second set of notes here over mitosis. We left off at how we're controlling the cell cycle, so let's jump back into it here. When we want to control a cell cycle, most of our differentiated human cells are in the G1 phase. Some types won't ever progress past that stage. They will just continue doing their job and be in a cell for the rest of your life. So if a cell is going to divide, something that's dividing all the time, perhaps like hair, root, or skin cells, that's determined by gene expression. So gene expression is what genes are turned on and what genes are turned off. So some of these genes will cause the cell cycle to advance. Other genes you can express to stop the cell cycle. And so we have built-in checkpoints within the DNA that tells a cell whether it should divide or whether it should just hang out for a while. And so these built-in checkpoints allow, hopefully, enough time for us to correct any problems we might have in our cell before we proceed with division. So when a nucleus divides by mitosis, each new nucleus has the same chromosome number as the parent cell. We've already said they're clones. Quick review, what are the 4 stages of mitosis? You need to know this by now. Hopefully you quickly rattled off prophase, metaphase, anaphase, telophase, PMAT, because you need that. So when we're going to go through mitosis, our cell's chromosomes in interphase, you can't even see them. In interphase, the chromosomes are super loose, and it just looks like a little black dot in a microscope. There's just DNA everywhere. And they need to be loosened up because we have to get some big old proteins in there in order to carry out transcription and in order to carry out DNA replication. Both of these are enzyme mediated. And so we have to have room to get these big clunky proteins in there to transcribe genes and to make copies of our DNA to go about being a cell. Your DNA needs to be loose in the nucleus. Now, when we're going to go through nuclear division, we can't have that. That loose DNA would get tangled up. It could be broken easily. And so what we're going to do when we go to prepare for mitosis is we're going to get these chromosomes really, really condensed and form this kind of X sort of shape that you're used to seeing in cartoons of mitosis. And so we get these chromosomes really tightly condensed about the centromere, and that tight condensation keeps them from getting tangled, keeps them from becoming broken, and keeps that chromosome together during mitosis and for being drug around by microtubules. And so here's what interphase would look like. Interphase, this is a nucleus. You can't really tell anything in that nucleus. Like, what does that even look like? I don't know. Just there's purple stuff everywhere. You can't really see much. And so interphase cells, interphase is not really part of mitosis, but the nuclear envelope is intact, and there's a ton of DNA in there, and that's really all we can see. So this is what it'd look like under the microscope for a plant. This would look like for an animal. Now, when we start to get into prophase and mitosis is officially beginning, we stop transcribing messenger RNA. You cannot be making messenger RNA and proteins at the same time you are dragging chromosomes around. So we got to stop what they're doing, and the DNA is going to start to condense. And when it does, you're going to see light through here. And so when you see these areas where there's light, and there's areas where there's little dense DNA, that's prophase or early prophase. And we still have that little nucleolus in there too, which is kind of cool. So the nuclear envelope is going to break up, and our centrosomes are going to become duplicated because we're going to need these little guys right here to organize our microtubules for us and drag our chromosomes where they need to be. So that's prophase. No nuclear envelope, dense chromosomes, centrosomes duplicating. So what happens during prophase? Alright, let's see how close we were, huh? Chromosomes condense, check. Microtubules form a spindle. Our nuclear envelope breaks up, check. Microtubules attached to chromosomes, check. Okay, so we've got a lot going on already. Centrosomes, these little guys right here, these two orange fellows. Centrosomes are a region near the nucleus that are going to organize our spindle microtubules. You could say the spindle grows from that centrosome. And they normally include two centrioles. Centrioles are the area where the microtubule actually grows out from and assembles from. So that's kind of cool. So here we have a cell that is truly in prophase. It's not early prophase. This is certainly prophase. Nuclear envelope, gone. Okay. Very condensed chromosomes. And here we have a little cooler view of these centrioles, or centrosomes, probably. Centrioles are rather small. And a little bit of spindles starting to grow out of these. That's kind of that fuzzy purple area. Not a lot, but a little bit. And some very condensed, wiggly purple lines that is the DNA. And so that's kind of good. Generally, R2, let's go ahead and write this down for you. R2 centrosomes are going to move to the opposite sides of the cell. And so here you have one up here, and you have one down here. Because at the end, we got to drag half of our chromosomes that way, and the other half of our chromosomes that way. And so our spindle microtubules are going to just start to assemble and bind to chromosomes around the centromere. Centromere is what hooks the middle of two sister chromatids together. So, the spindle. I'll take a minute and talk just about how cool these little microtubules are. A dynamic network of microtubules that forms during nuclear division is what we call spindle or spindle fibers. They do grow into the cytoplasm from opposite poles of the cell, and they attach to the duplicated chromosomes. They are made of microtubules, and they attach to different sister chromatids, and their job is to separate them. You want spindle fibers to find each chromosome. You don't want them to break. You don't want them to lose any chromosomes. That would all be very bad. So spindles are incredibly important in this process. Here's what they look like. Oh, so cool. Man, thank goodness for transmission electron microscopy, am I right? I know they added this color and that's fake, but it still looks great. Chromosomes, it's just a mess in here. Look at that. There's just DNA everywhere. And each one of these little spindle fibers has found the centromere, between two sister chromatids, and has attached to them. And as these are going to shorten, this end here will depolymerize. As they will shorten, they will pull one copy this direction, one copy this direction. So we call this the pull. Looks like one of them's already made it, or it was lost along the way. That'd be bad. But somebody's over there already. And so we call these the pull, right? Just like north to south pole of the planet, right? We got two poles of the cell, and these poles are where our centrosomes are and where our spindle fibers are growing from. So very cool picture. All right, metaphase and anaphase. Metaphase, that's this guy. Think M, middle, A, apart. Metaphase, all of our duplicated chromosomes line up midway between the spindle poles. You can call it a metaphyseal plate if you want to be really nerdy about it, or you can just call it the middle if you want people to understand you. So the metaphase, they just line up in the middle. Anaphase, think apart. Microtubules actually pull these sister chromatids from each chromosome to the opposite spindle pole. So one copy goes one way, one copy goes the other way. So here's what they look like. Again, we're comparing a plant cell with an animal cell. Plant cell, metaphase, they're all lined up in the middle. Animal cell, they're all lined up in the middle. Very visible centrosomes, very visible spindle fibers. Very cool. Anaphase, this might be the easiest to see under the microscope, especially in plants. Here we've got one set of each sister chromatid heading this direction, one set of each sister chromatid heading this direction. We got the same thing in the animal cell. They're getting split up right here, being pulled by those spindle fibers. And so that is, once they're in anaphase, we got to change our naming just a little bit. We can't really call them sister chromatids anymore. Once they're pulled apart, we can go back to calling them just a regular old chromosome. So we've left that cool little characteristic X sort of figure. We now have just sort of a straight line. One line from that blue X went this way. One line from that blue X went this way. And we've separated them. So that takes us to telophase. Telophase, some folks say telophase, pick your favorite. Two clusters of chromosomes reach our spindle poles, and we are going to go ahead and make that nuclear envelope. We took it away in prophase. We'll bring it back in telophase around each new cluster of nuclear material. We have 2 new nuclei now. Each has the same chromosome number as the parent cell. And we have a clone. 2 clones, actually. Pardon me. So here we have 2 nuclei. They're not real pretty yet, but they're there. Here we have 2 new nuclei with their nuclear envelopes around them. And this one's actually already started to do some cytokinesis. You can see it kind of pinching in there. That's kind of cool. So that's how these 2 cells are dividing. So now we got to talk cytokinesis, which is splitting a cytoplasm. Most of our eukaryotes, the cytoplasm is going to start to divide about late anaphase, and it's going to finish up the end of telophase. But the mechanisms can differ quite a bit between plants and animals, because in animals it's pretty simple. You just pinch the membrane in half. Plants is pretty tricky because you got to build cell wall. So cytokinesis is the process of cytoplasmic division. And in animal cells, we are going to form what is called a cleavage furrow, as these little actin filaments start to make a band around the middle of the cell and pull that band in. They contract to pinch that cell into 2 cells. And when they do that, they make this band of infolding in the cell membrane, and we call that a cleavage furrow. Once that cleavage furrow penetrates completely through the cell membrane, we have 2 new cells, and that's all there is to it. Plant cells are trickier because we have to form a cell plate. Cell plate is this big cellulose structure that's going to make our cell wall between the 2 neighboring cells. Midway between the spindle poles is where this is going to occur, and it's going to partition our cytoplasm once it reaches the parent cell wall. And so we got to divide these 2 with this big tough cellulose cell wall, leave some little plasmodesmata in it so they can still exchange ions, but that's the way we're going to do that. And so here's the animal cell, 2 distinct nuclei and telophase rather, and then cytokinesis. This little actin band starts to contract in, pinches in, and then we have 2 cells. Plants, we have let's jump down to here, I'd say probably telophase. We have that nuclear envelope. We get a whole bunch of little vesicles that are going to form this cell plate. The cell plate starts to grow outward, and once it divides these 2 cells completely, then we're done with cytokinesis for plants. So telomeres are a really cool piece of the whole mitosis story, because at the end of our DNA strands, we get these non-coding DNA sequences, and we call them telomeres. And they didn't get a lot of attention for a long time because they were DNA that didn't make protein. And for the longest time, we thought, well, if DNA doesn't make protein, then it must not be that important. But we're finding that that's not necessarily the case. Telomeres protect our eukaryotic chromosomes from losing genetic information. And what we get is our vertebrate telomeres, so animals that have a spine, have a short DNA sequence, and it goes TTA-GGGG, and it is repeated thousands and thousands and thousands of times at the end of your chromosomes. Each time you go through mitosis, you lose a little piece of that telomere, and it becomes shorter and shorter and shorter and shorter, and eventually the telomere is gone. And when your cell goes to check and make sure all the DNA is there for that chromosome, and it finds that you have no telomere, we basically get the signal that says, no, we're not ready to divide. And we're not ever going to be ready to divide because the telomere is gone. And so it's time for that cell to die. And so shortening telomeres are associated with aging. I would not be so bold as to go so far and say they're the cause of aging by any means, but certainly they are one of the cues that tells a cell to die. And they give us something of a clock. We can look at how long the telomere is and give a ballpark number of about how many more times it can divide before that telomere is going to be gone. And so it's important that to understand their role in division and in controlling how old a cell can really become. And so telomeres, of course, they're very important because our polymerases can't copy the last 100 or so bases. And when telomeres get too short, we don't pass our checkpoints, and the cell dies. So there's a certain number of times the cell is going to divide before it's going to die. So now there's an enzyme called telomerase, which affects telomeres. And in an adult, when we have stem cells, these stem cells can divide indefinitely. And one of the reasons they're immortal is because they have maintained this ability to make this really cool enzyme called telomerase, and that reverses telomere shortening. It adds those bases onto the end of telomeres, and so that these cells can divide and divide and divide and divide, and essentially never get old. And so research is kind of suggesting that this built-in limit on our cell division could be part of the mechanism that sets our lifespan. And of course, the question would be, could we add telomerase to other cells and cause them not to get old and die? Maybe we could. Would that be good or bad, or would we just be passing on, additional mistakes in the DNA? I don't know. That's kind of a tough thing to answer, but there's an enzyme that basically undoes cellular aging from the DNA perspective. So that's kind of a cool thing to sort of think about. Here they've illustrated telomeres. It's nothing that glorious really. Chromosome, telomere, 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 telomere. That's about it. They're the ends of chromosomes. They're cool. I guess. Why not? There's some colorful telomere Christmas lights for you. All right, cool. So if enough of our checkpoints fail and a cell loses control over its cell cycle, then we can form some bad stuff. Cell cycle's there for a reason. We need it there. If it's not there, we can get a neoplasm. Neo means new. It's a group of cells that have lost control over how they grow and divide, which means they're going to divide whenever they want to, which is bad. If a neoplasm forms a lump, we call that lump a tumor, and people start to freak out a little bit, which is understandable because we have a big mass of cells that is not obeying the orders to stop dividing, and that can very well become cancerous, and that's not good. So there's a few things that we think are causing these. We have pretty good evidence to support this, and one of these is called an oncogene. Onco means cancer. If you're an oncologist, you are a cancer doctor. So if you're an oncogene, you are a cancer gene. And that is any gene that will help transform a normal cell into a tumor cell. Not a very good thing to hear about. These genes will encode proteins that promote mitosis. They cause more mitosis. We call those proto-oncogenes, which means you are before the oncogene. You're not a gene that will cause cancer yet, but you could easily become one in the future if you become mutated. And so growth factors are another thing that can cause this. Growth factors are molecules that stimulate a cell to divide and differentiate. And one of our genes that encodes epidermal growth factor is an example of a proto-oncogene. As long as it's not mutated, the epidermis grows, everything's cool. If it becomes mutated, cells in the epidermis, your skin, grow out of control, and that's not cool. We call that cancer. Not good. And so here's a picture of the effects of these oncogenes and the overactive EGF receptors, where we have unregulated growth occurring in skin cells. And that is a bad deal. So just like there are genes that can cause cancer, there are also genes that can slow or stop cancer. And so there are checkpoint gene products that inhibit mitosis, and we call these gene products tumor suppressors. If the tumor suppressors are there and abundant, they are going to make cells not divide when cells are supposed to not divide, and everybody's going to be cool. If these tumor suppressors are missing, that can be a bad thing. So some examples of this, the products of BRCA1 and BRCA2, these are both genes, and these genes are examples of tumor suppressors. They regulate the expression of DNA repair enzymes. So this is kind of a lot to take in, but if this gene is working and this gene is working, they code for proteins that become DNA repair enzymes. The DNA repair enzymes fix errors in your DNA, and if your DNA is not being mutated, or rather is being repaired, even though it's being mutated or mistakes are being made, you are going to be combating the probability of having cancer. If these two are not being expressed abundantly, then you're not suppressing these tumors, and your likelihood of developing a tumor and cancer are significantly increased. So, the bad news is some viruses, such as human papillomavirus, HPV, which we're vaccinating people for pretty frequently now, can cause cells to make proteins that interfere with tumor suppressors. So if you're stopping the tumor suppressor, you are making cancer much more likely. And the last thing you want is a virus that's going to increase your chances of cancer. Not fun. So here's some checkpoint genes in action. And here's some terms for cancer. Benign and malignant. You want to know these terms. Benign, if you've got to have something, you'd rather hear benign than malignant. Benign neoplasms, a new growth that is benign. Like an ordinary skin mole, they'll grow, but pretty slow. They're really going to stay in one place, and these things are generally not cancerous. So that is good. Malignant neoplasms. Malignant is a bad word. That means it's cancer. It's going to disrupt body tissues. A lot of times it can move around, affect other body tissues, change our metabolism, get progressively worse, dangerous to your health, not good stuff. Cancer is causing somewhere between the order of 15 to 20% of all human deaths in developed countries. And so malignant neoplasms are cancers, and that is not a good thing. So how do you know a cancer cell is a cancer cell? Well, if it grows and divides abnormally, that's probably a cancer cell. Capillary blood supplies to the cells may increase abnormally, which means cancer cells know how to feed themselves. They will create products that bring blood vessels to the cancer cell. It's called angiogenesis, to feed the cancer cell and feed the tumor. And the cytoplasm and the plasma membrane are going to be altered. So these malignant cells typically have an abnormal chromosome number. The metabolism might shift them toward fermentation instead of aerobic respiration, and they might be missing proteins that will impair the function of that plasma membrane. And so lots of mistakes in the genetic material makes lots of mistakes in the ending cell product. So the other thing is cancer cells can break loose and invade other parts of the body. If this happens, it's called a metastasis. This is another really bad word. Words that start with M are bad. Malignant's bad. Metastasis is bad. Basically, it means a piece of this tumor can break off. Can float around through your lymphatic, blood vessel, something like that, attach in a new place, and give you cancer in that place. Not good. Due to some altered recognition